In today's video, I want to talk about regulation of the gene expression. We all know that our organisms consist of many different cells and they're very different from each other. Um, so what makes them so different? Let's take, for example, a nerve cell and say a muscle cell. They're very different, but um, actually composition of each cell, genetic composition, is totally the same. So what is different is a gene expression. In nerve cell, some genes is turn it on and some turn it off, and different genes in muscle cell turn it on and off. So that's why they look so different, and they serve for different purposes. So if we take piece of genome, I will draw a line. So it would be just representation of the piece of the genome, and let's say this box here would be representation of the gene. So there is sequences that lay upstream from the gene that regulate expression of this gene, and one sequence, call it promoter, and usually it lays it's also called -35 region because it lays 35 nucleotides upstream from the first nucleotide of the gene, and it is usually in eukaryotic cells start codon IUG, and it's also called 0 point. So this is -35 region. As I said, it is a promoter where different proteins bind to this region in order to promote gene expression. But also upstream we can find different control sequences that also influence gene expression. So we can call promoter as switch that have two positions like on and off, and control sequences, they are more like resistor that makes lamp bulb to shine brighter or dimmer depending on the way which we turn, making resistance more or smaller. So control sequences is sequences to on the genome where proteins bind. So as I said, promoter is mostly like switch with on and off position, and control sequences is can work two ways. They can promote gene expression, or they can suppress gene expression. Um, so this is simplified representation of how regulation of the gene works. Thank you for your attention. Goodbye.